Happy Sunday, everybody. I'm Dr. Gregory, and you're watching Dr. Gregory's Meta Moments. This is episode 4. I had so much fun talking about the biology of belief last week by Dr. Bruce Lipton that I decided to go ahead and head on to chapter 2. Chapter 2 happens to be a little more pointy-headed, a little more based on scientific studies and the description of DNA and how proteins are built. But I'm going to do my best to explain to you what I read through the chapters so you can get a better idea. The chapter's name is pretty funny. It's called It's the Environment, Stupid. And where he gets this is, my professor, mentor, and consummate scientist, Irv Koenigsberg, was one of the first cell biologists to master the art of cloning stem cells. He told me when cultured cells you are studying are ailing, you first look at the cell's environment, not to the cell itself for the cause. So an analogy was Bill Clinton's campaign manager, James Carville, who decreed, "It's the economy, stupid," for the mantra of the 1992 presidential election. So Bruce Lipton decided to change this a little over to, "It's the environment, stupid," and we're going to get into why. Since the dawning of the age of genetics, we've been programmed to accept that we are subservient to the power of our genes. The world is filled with people who live in constant fear that on some unsuspecting day, their genes are going to turn on them. Consider the masses of people who think they're ticking time bombs. They wait for cancer to explode in their lives as it exploded in the life of their mother or sister or aunt or uncle. Millions of others attribute their failing health not to a combination of mental, physical, emotional, and spiritual causes, but simply to the inadequacies of their body's biochemical mechanics. Of course, there's no doubt that some diseases, like Huntington's chorea, thalassemia, and cystic fibrosis, can be blamed entirely on one faulty gene. But single gene disorders affect less than 2% of the population. That's mind-blowing. That means that when you are concerned about genetic disorders that are potentially passed on to you, the likelihood of that being the absolute cause of your disease process is less than the factors like mental, physical, emotional, and spiritual causes. Only 2% of genetic diseases are actually passed on completely and totally hereditarily without much influence from the environment. That's pretty interesting. So, diseases that are today's scourges like diabetes, heart disease, and cancer, they can short circuit a happy and healthy life. But they are not the result of a single gene. They're the result of complex interactions among multiple genes and environmental factors. So that's very important to know. So this is where the confusion comes. The media had repeatedly distorted the meaning of the words correlation and causation. And that's one thing to be linked to a disease, but it's quite another to cause a disease, which implies directing and controlling action. So if I show you my keys and a particular key controls my car, you might think this makes sense because you know you need that key to turn on the ignition. But does the key actually control the car? If it did, you couldn't leave the key in the car alone because it might just borrow your car for a joyride when you're not paying attention. But really, in truth, the key is correlated with the control of the car. The person who turns the key is actually the one who controls the car. Specific genes are correlated with an organism's behavior and characteristics, but they are not activated until something triggers them. What's that trigger? It's the environment, stupid. So, what activates genes? Let's talk a little bit about that. The idea that genes control biology is a supposition. It has actually never been proven. In fact, it has been undermined by the latest scientific research. Genetic control has become a metaphor in our society. We want to believe genetic engineers are now the new medical magicians who can cure disease while they're at it and create more Einsteins and Mozarts, but metaphor does not equate with scientific truth. When a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates the expression of that gene. In other words, when it comes to genetic control, it's the environment. So, our cells are mainly an assembly of protein building blocks. One way of looking at our trillion-celled bodies is that they're protein machines. Although, as you know, I think we are more than machines. It sounds simple, but it isn't. So let's take one thing. It takes over 100,000 different types of proteins to run our body. And let's take a closer look at how our cells, the 100,000 proteins, are actually assembled. Each protein is a linear string of linked amino acid molecules, like this. So we're giving the example, my poor puppy. We're giving the example of a pop bead analogy because everybody's pretty familiar with it, but it's not the exact replica of how an amino acid works because an amino acid proteins create a slightly different shape. So to be completely accurate, think of a pop bead necklace that got mangled in the factory, so it's more clumped up in nature. There are 20 amino acids which comprise the protein backbone, okay? And they have a unique shape and conformation. And consider the differences between the character of the backbone that's made by identically shaped beads and the one assembled from the differently shaped fittings, such as in this illustration above. The backbone versus the beads themselves. Most amino acids have positive or negative charges, which act like magnets. Like charges cause the molecules to repel each other. So two positive charges would repel those beads apart from one another. But opposite charges would bring them together. They would attract each other. Like this. So you can see that the backbone is made up of the same proteins, but the way the actual protein itself is arranged is based on the charges and what attracts and repels one another. That actually changes the shape of the protein itself. Since positive and negative charges attract one another, the backbone of the amino acids will rotate around their bonds so that the positive and negative terminals will come closer together. So it shows how you can change from different conformations simply by changing those amino acids and the protein and the charges associated with them. Changing conformation generates movement. And the movement is harnessed to do work, providing for such functions as digestion, respiration, muscle contraction. So when that signal molecule detaches, the protein returns to its preferred extended conformation. So this is how a signal-generated protein movement provides for life. The signal comes in, it has an attraction or repellent to it based on its charge, and it makes the protein move in such a way that it allows for certain bodily functions, and when that signal is removed, the protein goes back to its original state. Pretty interesting, huh? So this would be an example of different proteins, different conformations. However, we're using the same building blocks, just like Legos, right? Except now we're rearranging them in such a way that they look different and perform completely different functions. You'll notice that we didn't discuss DNA at all. It's because it's the changing of the protein's electromagnetic charges that's responsible for behavior-generating movement, not DNA. How did we get to the widespread and cited notion that genes control biology? In The Origin of Species, Darwin suggested hereditary factors were passed on from generation to generation and controlled the traits of the offspring. His influence was so great, scientists myopically focused on identifying that hereditary material, which they thought controlled life. So in 1910, intensive microscopic analysis revealed hereditary information was passed on generation after generation, and it was contained in chromosomes, thread-like structures that become visible in the cell just before it divides into two daughter cells. So chromosomes are incorporated into the daughter cell's largest organelle, which is a smaller portion of the cell, called the nucleus. When scientists isolated the nucleus, they dissected the chromosomes and found the hereditary elements, which are comprised of two kinds of molecules, protein and DNA. Somehow, the protein machinery of life was entangled in the structure and function of these chromosome molecules. The understanding of the chromosome's function was further refined when Watson and Crick showed that DNA was what actually contained hereditary information. So, DNA is basically made of 4 nitrogen-containing chemicals. They're called bases, and these 4 bases are adenine, thymine, cytosine, and guanine. They're abbreviated as A, T, C, and G. The DNA spells out a sequence of amino acids along the protein's backbone, and these long strings of DNA molecules can be divided into single genes, which are segments that provide the blueprint for specific protein backbones. So the code for recreating the protein machinery had been cracked. But the suggestion that DNA controls its own replication and serves as the blueprint for the body proteins led them to create this central dogma. The belief that DNA rules. The dogma was so fundamental to modern biology, it was essentially written in stone. And the equivalent of science's 10 commandments, the dogma refers to as the primacy of DNA, and it's a fixture of almost every scientific text. Now, the scheme of life unfolds. DNA goes to RNA, and RNA is basically a short-lived Xerox copy of DNA, and such, a physical template which encodes the amino acid sequence makes up the protein's backbone. The central dogma's assumption is that there is a one-way flow of information from DNA to RNA to protein. That's profoundly important in this concept that was created many, many years ago. Since proteins represent the physical body, the dogma implies that your physical body and your life experiences cannot send information back and alter the DNA. So what that means, would have meant is that according to the dogma, DNA controls your life and you cannot influence your DNA. That's not true. This was what was originally thought, but it's not true. So we enter into the Human Genome Project many years later. It was a global scientific effort started in the 80s. And from the outset, it was an ambitious one. Conventionally, we thought that the body needed one gene to provide the blueprint for each of the 100,000 different proteins that make up our body. And that at least 20,000 regulatory genes, which orchestrate the activity of protein encoding genes. Then scientists concluded that the human genome had to contain a minimum of 120,000 genes to create these 23 pairs of human chromosomes. Geneticists experienced a considerable shock when, contrary to their expectations of how many genes humans actually have, they found that the human genome actually only contains fewer than 25,000 genes. Much, much less than they originally thought. So, unless a human genome contains a lot of genes that are opaque to our computers, it's clear we do not gain our undoubted complexity over worms and plants by just using more genes. Because there are a lot of organisms that have as many genes, if not more than we do, yet are less complex in their organization, less complex organisms. So now we can't use genes to explain why humans are at the top of the evolutionary ladder. There's not much difference in the total number of genes found in humans and those found in primitive organisms. Here's an example. A primitive Cinorbitus worm serves as a perfect model for studying the role of genes in development. It's a rapidly growing and reproducing organism, and it was patterned of 969 cells with a simple brain of about 302 cells. However, this genome actually consists of 24,000 genes in that worm. The human body is comprised of 50 trillion cells, yet it only has 1,000 more genes than this lowly, spineless, 1,000-celled microscopic worm. Interesting, huh? So here's another question. This is what we also were taught in medical school and in science. Is the nucleus truly the cell's brain? If our assumption that the nucleus and its DNA-containing material is the brain of the cell, then removing the nucleus which is called enucleation, would result in immediate death of the cell, wouldn't it? Well, now for the big experiment. That's exactly what they did. They took micropipettes and removed nucleus from cells. Guess what? Following enucleation, many cells survived for up to 2 or more months without genes. They were viable enucleated cells, and they do not lie around like brain dead lumps of cytoplasm. They were actively ingesting and metabolizing food. They maintained coordinated operations of their physiologic systems. They retained their ability to communicate with other cells. They were able to engage in appropriate responses to growth and protection requiring environmental stimuli. But enucleation was not without side effects, of course. Without their genes, they were not able to divide. So they were not able to produce any protein parts that they would have lost through normal wear and tear of the cytoplasm. So, because of their inability to replace these small proteins, it would contribute to the mechanical dysfunction of and ultimately result in the death of the cell. Because they would have parts that would eventually wear out, and they didn't have the nucleus or the genetic code to be able to replace those proteins and keep the cell healthy and moving along. It's kind of like similar to oxidative damage in your body, and how your human body replaces those damaged cells with new cells, which happens every few days in your stomach lining, for instance, believe it or not. Our experiment was designed to test the idea that the nucleus is the brain of the cell. But if the cell had died immediately following enucleation, the observations would have at least supported the belief. But the results were unambiguous. Enucleated cells still exhibited a complex, coordinated, life-sustaining behavior, and implies that the cell's brain is still intact and functioning. If the nucleus and its genes are not the cell's brain, what exactly is the DNA's contribution to a cell? If the enucleated cells die, not because they've lost their brain, but because they've lost their ability to reproduce, without the ability to reproduce parts, enucleated cells cannot replace failed protein building blocks, nor replicate themselves. So, the nucleus isn't the brain of the cell, it's the gonads. It's the reproductive organ of the cell, not the brain. Huge information. So the science of epigenetics, which means control above genetics, profoundly changes our understanding of how life can be controlled. And in the last decade, epigenetic research established that DNA blueprints passed down through genes are not set in concrete at birth. So genes are not destiny. Environmental influences, nutrition, stress, emotions, all of these things can modify your genes without changing the blueprint. How does that happen? It's a process. I'm going to explain this to you very easily. And he uses a really good analogy of a television set. Here's another interesting thing. When they took out the DNA, they studied the DNA, and most of the scientists threw away the proteins thinking that was junk, that had no data involved. It was like throwing the baby out with the bathwater. But now epigeneticists are bringing back the baby and studying the chromosomes proteins, because proteins are turning out to play a very crucial role, as crucial as the heredity of DNA. The DNA forms the core. The proteins cover the DNA like a sleeve. So when genes are covered, their information cannot be read. Imagine your bare arm as a piece of DNA representing the gene that codes for blue eyes. But in the nucleus, this stretch of DNA is covered by the bound regulatory proteins, which cover your blue-eye gene like a shirt sleeve, making it impossible to read. How do you get this sleeve off? You need an environmental signal to spur the sleeve protein to change shape and detach from the DNA such that the gene can be read, such that the sleeve can be pulled back. Once the DNA is uncovered, the cell makes a copy of that exposed gene, and as a result, the activity of the gene is now controlled by the presence or absence of these sleeve proteins, which are now in turn controlled by environmental signals. So, back to this analogy of a test screen I was telling you. Think of the pattern of a test screen as the pattern encoded by a given gene, say one for brown eyes. The dials and switches of the TV fine-tune the test screen by allowing you to turn it on and off, modulating characteristics like volume, color, hue, contrast, brightness. By adjusting the dials, you can alter the appearance of the pattern on the screen, but you're not actually changing the broadcast pattern. That's the key there. This is precisely the role of regulatory proteins. So this would be, the dials are like the regulatory proteins. The signal is the DNA. It hasn't changed, the broadcast signal. But the regulatory proteins, which are the dials where you can change the hue, the color, look how it changes the actual image or the final result. It makes quite a difference. So, another example that they gave is something where they bred agouti mice, and the dietary supplements were given to pregnant mice that had this abnormal agouti gene. And I'm about to show you these mice. The agouti mice have yellow coats, they're very obese, and it predisposes them to cardiovascular disease, diabetes, and cancer. So these two mice are sisters. The one on the right is the yellow agouti mouse, which is predisposed to a lot of illnesses. The one on the left is well, I guess it's on your right, is the normal mouse without the agouti gene being expressed. So in the experiment, one group of agouti mice received supplements like folic acid, B12, betaine, and choline. These are methyl-rich supplements. The mothers who received these methyl-rich supplements, guess what? Their offspring were standard, lean, brown mice, and even their offspring, even though their gene was the same, quote, agouti gene as their mother's, the agouti mothers who didn't get the supplement produced yellow pups just like the moms, which ate much more than the brown pups. So think about it. Agouti mice, the yellow ones, the ones predisposed to disease, one group of pregnant mice were not fed these methyl-rich supplements, one group was. So they had the same genetic blueprints, right? But guess what happened when you change the environment of one set of mice? The offspring of that mice had normal offspring with no agouti tendencies, whereas the ones that were not fed the supplements looked just like their parents, or basically they replicated the same patterns of disease. Super interesting. We can talk a little bit more about also that DNA does not control biology, and the nucleus itself is not the brain of the cell. So just like you and me, cells are shaped by where they live. And in other words, it's the environment, stupid. Okay, so lastly, we talk about dark DNA. There was a portion of DNA that was found when they were doing studies on DNA, and they thought it was junk DNA. They actually called it junk DNA because they didn't see that it was encoding any specific biological functions in the body. So they would actually remove this junk DNA and studied only what they thought was the active DNA, which is quite interesting. And we're trying to reterm this phrase from junk DNA to dark DNA because dark DNA just implies that there is potentially functions, but we just don't know yet. So it's not useless DNA. We'd rather term it dark DNA. Dark DNA uses epigenetic mechanisms that enable the human cell with 19,000 gene blueprints to code for over 100,000 different protein molecules. So that goes back to answer that question. How is it that we only have 25,000 genes, quite similar to a very underdeveloped worm, with lower on the evolutionary totem pole, if you will, yet we have all of these proteins we have to make? Well, guess what? We do it because we have dark DNA that is controlled by epigenetics, meaning the environment, the protein, and hence the gene gets read. And that's how we can make so many different proteins in our body, and that's how we became so complex as human beings in our bodies. Pretty awesome. It's pretty much like I said before, like using an erector set or Lego blocks. You have the same building blocks, but you're arranging them in different ways. The body plans for each organism, which is encoded in the dark DNA, and they're directly connected to the dynamic environment with epigenetic mechanisms. So it can interpret, translate, and control the activity of these protein encoding genes. Let's also talk a little bit about telomeres. Telomeres are kind of like the endpoints of DNA. They're like as if you had laces with an aglet, okay? So these are extensions and they prevent the DNA from unwinding. They're kind of like a block there. They're very necessary in our bodies because they resemble like the plastic tips of the shoelace, like I said, but if it fails, then the woven threads in the tip of the shoelace could unwind and become frayed, which could impair the function. And also, telomeres provide a physical platform for DNA to replicate. So when DNA replicates, instead of trying to replicate over the same sequence over and over again and potentially fraying the ends of it, the telomere allows for the replication to occur all the way into this aglet, if you will, this protective area. So even if a piece of the telomere came off, it would be okay because at least it's not fraying the DNA signal itself. So the longer the telomere, the more survival effect it has, if you will. They basically prevent the loss of protein encoding information during gene replication. So they are basically our armor, if you will. And now you're going to get depressed about how your stem cells are eventually going to divide and die, but we have great news. We identified a special enzyme called telomerase whose function is to extend the telomere length. So telomerase activity is the molecular equivalent of a fountain of youth because it replenishes these telomeres, these protective ends of the DNA, and increase the vitality and reproducibility of the cells. Telomerase activity enhances health and extends life. That's exciting news. So here's the catch. Did you know that life experiences can actually stimulate or suppress telomerase activity? So stressful prenatal development, childhood abuse, domestic violence, PTSD, nutritional disabilities, lack of love, these can all inhibit telomerase activity. That's a big deal. These factors contribute to the onset of disease and a shortened lifespan. In contrast, exercise, good nutrition, a positive outlook on life, living in happiness and gratitude, being in service, experiencing love, these all enhance telomerase activity, the fountain of youth. So we can consciously empower ourselves by actively embracing our own telomerase. And yes, because I can't repeat this enough, factoring in all the wonderful research that has been done in the last decade, it's the environment, stupid. Thanks for watching. I know that was a tough one, you guys, but if you need a little bit more understanding and if you need me to explain a little bit more, I can always send messages and we can talk a little more, whatever you need. But thanks for watching. Happy Sunday. Love you all.